Good afternoon and a very warm welcome to all. So we can discuss the continuation of the important culture media which are used in the microbiology laboratory. So next culture media is his serum water medium. His serum water medium, it is used in carrying out fermentation tests with certain pathogenic organisms such as pneumococcus, which is the causative agent of pneumonia, that do not grow well in the peptone water. So, in the peptone water, normally pneumococcus doesn't grow well. So, we are using, instead of peptone water, we are using his serum water. And in this medium, the fermentation takes place and it is indicated by the change in the color of the indicator by the production of acid. And that may also coagulate the serum. That acid coagulates the serum. This media is used to study the fermentation reactions of the bacteria, which cannot grow in the peptone water sugar media. And this type of medium are used in pneumococcus, mainly pneumococcus, then Neisseria corynebacterium also. This is his serum sugar medium with serum enrichment. So, as a result of this acid production, it may coagulate the serum present in the media. Next is about Lowenstein-Jensen medium. Lowenstein-Jensen medium, which is also known as LJ medium, it is a selective medium that is commonly used for the cultivation and isolation of Mycobacterium, specifically Mycobacterium tuberculosis, from clinical specimens. LJ medium was originally formulated by Lowenstein, who incorporated Congo red and malachite green to inhibit unwanted bacteria growing in the media. And now the present formulation, it comprises of a glycerated egg-based medium is now used, and it is based upon Jensen's modification. So the name Lowenstein-Jensen media. And how the Jensen modified? The Jensen modified Lowenstein's medium by altering citrate and phosphate contents within the medium, eliminating the Congo red dye and increasing the malachite green concentration. That's why the media appear green in color. Gruft, another person, further modified LJ medium with the addition of penicillin, nalidixic acid and ribonucleic acid to increase selectivity. This medium supports the growth of a wide variety of mycobacteria, and can also be used for niacin testing. This is the composition of Lowenstein-Jensen medium. Potato flour, asparagine, monopotassium phosphate, magnesium citrate, malachite green, magnesium sulfate, glycerol and egg base. And the Lowenstein-Jensen Gruft modification is same as LJ medium, but addition of 3 ingredients, that is nalidixic acid, penicillin and RNA are incorporated along the medium. Egg is an enrichment material and it can stimulate the growth of tubercle bacilli. Malachite green inhibits the growth of the organisms other than Mycobacterium. Glycerol in the media promote the growth of Mycobacterium tuberculosis but not Mycobacterium bovis. So this is the cultural characteristics of Mycobacterium tuberculosis in the LJ media. And this is the LJ media, and the colony appears like this after 2 weeks. So the development of colony takes place so many times because of the longer generation time of the bacteria. Next is about Dubose medium. Dubose medium, it is recommended for the preparation of liquid medium for the rapid cultivation of pure cultures of Mycobacterium tuberculosis and related microorganisms. Dubose broth, it is formulated as per Dubose et al. And it is a modification of the medium originally formulated by Dubose and Davis and Dubose and Middlebrook. So, this medium contains tryptone L-asparagine as a source of nitrogen. Polysorbate 80, it is an oleic acid ester which acts as a surfactant. It therefore supplies essential fatty acids for the replication of mycobacterium, and it increases the growth by dispersing the bacilli. The phosphates together with calcium chloride buffers the medium as well as serve as a source of phosphates. Next is magnesium sulfate, zinc sulfate, copper sulfate and ferric ammonium citrate. They may provide trace metals and sulfates within the media. Bovine albumin binds the free fatty acids and which may be toxic to the mycobacterium. So albumin should be heat treated to inactivate the lipase activity, which may release fatty acids from polysorbate incorporated with the medium. In this medium, drug sensitivity of tubercle bacilli can be carried out, and it contains tween 80, that is bovine serum albumin, casein hydrolysate and asparagine and salts. Tween 80, it may causes dispersed growth, and bovine albumin causes rapid growth. So the contents within the medium. This is the Dubose medium. It is nothing but the liquid medium which promotes the growth of Mycobacterium tuberculosis. Next is Loeffler's serum or Loeffler's medium or Loeffler's serum slop. It is the modification of original formula developed by Loeffler in 1887. The Loeffler medium enhances primary and secondary isolation and cultivation of fastidious pathogenic microorganisms, especially from nose and throat. It also restores virulence and other identifying properties after they have been lost due to prolonged incubation or repeated subculturing. The high serum content helps in determining the proteolytic activity of the organisms. It is also used for the demonstration of pigmentation and ascospores. The media contains horse serum, beef extract, dextrose and proteose peptones, which together supply the complex nitrogenous substances and nutrients which are necessary to support the growth of Corynebacterium diphtheria. The sodium chloride, it is added to supply essential ions. Dextrose is a source of fermentable carbohydrate. The serum causes the medium to coagulate during the sterilization process and are sources of protein which are used for the metabolism of the Corynebacterium and other organisms. And the serum is used for enrichment for the increased multiplication of bacteria. Diphtheria bacilli grow in this medium within 6 hours when the secondary bacteria do not grow. It is used for the rapid diagnosis of diphtheria and to demonstrate volutin granules. It contains either sheep or ox or horse serum for the enrichment. Next is about Telluride blood agar. It is a selective medium for the isolation of Corynebacterium diphtheria. Telluride inhibits the growth of most secondary bacteria without an inhibitory effect on diphtheria bacilli. It is also an indicator medium as the diphtheria bacilli produce black colonies. The telluride metabolized in the medium get metabolized to tellurium, and which has and therefore produce grey to black colored colonies. Next is about Embiaga, Eosin methylene blue agar. Embiaga, it is a differential microbiological medium which slightly inhibits the growth of gram-positive bacteria and provides a color indicator distinguishing organisms that ferment lactose and those that do not ferment lactose. So the lactose fermenters are E. coli and non-lactose fermenters are Salmonella and Shigella. So to differentiate this lactose, both lactose and non-lactose fermenters, we can use Embiaga. It was originally devised by Holt Harris and Teague and further modified by Levin. It is thus a combination of Levin and Holt Harris and Teague formulae which aseptic digest of animal tissue and phosphate as recommended by Levin and 2 carbohydrates as suggested by Holt Harris and Teague. The medium is important in medical laboratories to distinguish gram-negative pathogenic microbes in a short period of time. So this is the appearance of the Embiaga. Lactose fermenters gives a metallic sheen, a green metallic sheen in the medium. So it helps to isolation and differentiation of enteric bacilli and gram-negative bacilli. And bacilli, those which are present in the intestine causing disease. It also helps in the isolation of lactose fermenting and non-lactose fermenting enteric bacilli. Next is about DCA. DCA means deoxycholate citrate agar. Deoxycholate citrate agar, it is a modification of Lowenstein's formula and is recommended for the isolation of Salmonella and Shigella species. So this medium recommended for the isolation of Salmonella and Shigella. This medium, it is similar to deoxycholate agar in comparison, but it is moderately more selective for enteric pathogens because of the increased concentration of both citrate and deoxycholate salts. Sodium deoxycholate at the pH 7.3 to 7.5, it is inhibitory for gram-positive bacteria. Citrate salts in the concentration included in the formulation are inhibitory to gram-positive bacteria and most other normal intestinal organisms. It is thus a selective and differential medium used commonly for the isolation of enteric pathogens. So it is the medium for the isolation of enteric pathogens. Next is tetrathionic broth. Tetrathionic medium, it is recommended as a selective enrichment broth for the isolation of Salmonella typhi and Salmonella from clinical specimens. The tetrathionic broth base APHA, it is enriched with iodine solution for the selective cultivation of Salmonella species. Bile salts are incorporated in this medium as a selective agent and it retards the growth of gram-negative bacilli other than Salmonella and also it inhibits gram-positive organisms. So these are all about tetrathionic broth. Rest of the medium we can discuss in the next class. Thank you.